second time an update for COVID-19, making sense of it all. This is a nonpartisan presentation. This carries in the tradition that we started well over a year ago, talking about tenant rights, talking about mental health during the pandemic. We hosted Police Chief Stephen Beecher, but tonight it really is important that we talk about COVID-19 in terms of the variants, the treatments, the vaccines, and also touching on misinformation. You'll see that Irene Sardonis is one of our hosts for this evening. She has put us all on mute. You just saw that this is being recorded. Dr. Sean will be talking for about 45 minutes or so, and we invite people to put their questions in the chat box during the presentation, and then we will get to the questions at the end of the meeting. We'll go for about, Dr. Sean will talk about 45 minutes. We'll get to questions, and we'll wrap up probably no later than, say, 8:15 at the absolute latest, given that it's a Monday night. So that being said, Dr. O'Connell, Dr. Sean OConnell, first and foremost, we're delighted that he is a resident of Mount Olive. And that alone makes him a smart guy, and I'm glad that he's with us here in town. But more importantly, Dr. O'Connell is a seasoned healthcare professional with 35 years experience in immunology, cell biology, regenerative medicine, and tissue repair. Dr. O'Connell is the founder and president of O'Connell Consultants. He has served as an interim management or strategic consultant to numerous venture capital, pharmaceutical, biotechnology, medical device, and tissue processing companies, focusing on operations, preclinical and clinical development, commercial opportunity assessment, product and market development strategy. His clients have spanned early stage startup through large cap companies. He's a chief scientist at AGIL Research and International Catalyst Partnership. Previously, Dr. O'Connell has served as chief science officer for Vertical Spine LLC and as chief medical officer for Cascade Medical Enterprises. As director, medical affairs for the transplantation, tissue engineering, and immunology business unit of Novartis Pharmaceuticals, Dr. O'Connell was responsible for preclinical and clinical development, publications, and professional relationships. He was also the senior vice president, medical director for Integrated Communications Corporation, and was the COO and co-founder of Heartland Biotechnologies, a drug discovery startup.
Can't seem to hold a job, right there, Andy?
I'm sorry I didn't memorize this. Dr. O'Connell receives his PhD in cancer immunology and molecular biology from Rutgers University. Dr. O'Connell has authored over 65 peer-reviewed publications and abstracts, and currently holds the position of adjunct professor in the Department of Vascular Surgery at Englewood Hospital and Medical Center, Mount Sinai School of Medicine, New York. Beyond that, when he's not out serving the scientific community, he plays guitar, and hopefully we'll have him back to do that one evening. But we're pleased, Dr. O'Connell, that you're here with us, and I'll hand the floor over to you. Thank you, sir.
So Irene, I got to make sure that I can get access to my PowerPoint here. Do I escape? How do I do that? Let me see. Aha. Can you see my screen?
No, sir, not yet.
All right, I've just gone full screen. Irene,
Okay.
what am I doing wrong?
Oh, David. Okay. Okay, this is somebody else hit share screen. Let me. Dave, did you hit share screen? Because it's saying, let's do this. Stop participant sharing. Okay. All right, Sean, on the bottom of your screen, there's a green button that says share screen, right?
1 second, I'm getting there. Yep.
Okay, so press that, and then pick your pick your presentation and then click the button again, share screen. And then your screen will be shared.
Can I do it from the desktop? Let's see.
Yeah, from your desktop.
Hope I get the right one.
Yes, yes, you did.
Good.
We see it now.
Excellent.
It's
Excellent.
we can see that.
All right, very good. Well, I want to thank everyone for joining us tonight. I know that some of you attended the first presentation, which was now some time ago. Today's will be more in the manner of an update, because while we're all now we're on 2 years now in this COVID, we're fairly familiar with the basics of it, but much has changed recently, and that's what I was really intending on talking about tonight. Let me go to full screen. All right, so my agenda, you should be seeing it up on the board. I'm going to talk about a couple of major topics. The first topic I'm going to talk about are the variants. In particular, I want to focus on Delta, because while there are certainly other variants, Delta in the United States is by far the main character. It's dominating almost all geographic areas within North America now. And then I want to talk about the boosters, which are very hot lately. Many I know here on this call have probably received their boosters. I've just scheduled mine. And then following that, I want to talk about vaccination in children, talk about some non-vaccine treatments that are, two of which have recently been approved by the FDA, talk a little bit about safety of vaccines, and then I'm going to introduce two other subjects toward the end. I'm going to briefly start to talk about long COVID. It's something we could talk all night about, but the fact is that there's still so much we don't know about long COVID, but I am going to talk about some of the things that we think are driving long COVID and what has to be done to alleviate people with the symptoms of long COVID. And then I am going to end with some interesting misconceptions and fake facts that have emerged recently about vaccines in particular. All right, so the variants. All right, the original SARS-CoV virus was identified in China in the fall of 2019. All current vaccines approved in the United States are based on that strain, SARS-CoV-2. All right. However, we know that during viral spread, as the virus spreads and infects more people, mutations arise. These are errors in copying of the virus's genetic information. They occur constantly. Most of these mutations have no effect or are harmful to the virus, in which case they disappear immediately. However, some can cause an advantage, can represent an advantage to the virus. They can either increase the transmissibility of the virus, which is definitely what we see in several of the new variants. They can increase its morbidity, its ability to make us sick, the ability to kill us, its mortality, and they can have decreased susceptibility to antiviral drugs, vaccines, natural immunity, as well as elude diagnostic testing. All right, so the original strain, it's got about 30,000 RNA letters, 30,000 base pairs, and this tells the virus, or tells the infected cell exactly how to make the virus, how to turn that cell, that infected cell, into a virus-making factory. During that process of replication, mistakes happen. And in the case of RNA viruses, they happen a lot more. And these would include other viruses like influenza and HIV. They occur much more frequently than viruses that use DNA as their genetic information. All right. So as I said, the original COVID-2 strain came along in the fall of 2019. Alpha came along shortly after, early in 2020, and it came from England. And it turned out to be quite a bit more infective, more transmissible than the original SARS. How? About 50% more contagious. And it quickly dominated not only the UK and Europe, but it quickly dominated the United States as well. Other variants soon followed Alpha in the course of 2020. These would be Beta and Gamma. All right. So this is just a sequence that shows Alpha. And why did it become more infective and spread more faster? All right. Essentially changes in the spike protein. The way that the virus binds to the cell. Alpha is much more efficient at binding to cells than the original strain of virus, the original Alpha. But the good news is that the vaccines are still very effective against Alpha, greater than 75%. It does have a somewhat higher incidence of death, but it's got a much faster doubling time. That's Alpha. All right. Then we had Beta and Gamma, and I won't dwell too much on these. They came essentially from South Africa and Brazil. What was bad news about these strains is they really knew how to elude the immune system. They developed mutations that, while in some cases increased transmissibility, what they really did was they caused changes in the virus that eluded antibody responses. Gamma, for example, which came out of Brazil, is probably the most lethal strain of all the viruses. That is, upon infectivity, you're going to expect the most severe disease and death rates associated with Gamma. All right. But then earlier this year, along came Delta and Delta Plus. This is a graph that basically shows the prevalence of a couple of the major strains. Delta is in orange. Alpha is in blue. This is Gamma. This is, I believe, Delta. I'm not Delta. This is Beta here. And what you can see is the time frame is very important. This is in end of May to the end of August of this year. And you can rapidly see what happens when Delta got into the United States. Within the space of 3 or so months, Delta took over. Such to the point that even now, you really don't see a lot of the other strains in the United States. And the reason is Delta is just so much more effective at transmission from one person to the other in a different league. And I'm going to talk about that in a minute. All right. Delta is 50% more contagious than Alpha. Alpha is 50% more contagious than the original strain. So you got a 100-fold increase right there. The infectivity of Delta is similar to chickenpox. What does that mean? All right. You'll hear the epidemiologists speak of something called R0, the R value, which stands for reproducibility or reproduction factor. And what it essentially means is if you take a population that is unvaccinated, hasn't been exposed to that virus, and you put an infected individual into that population, on average, how many other individuals are liable to contract the disease from that individual? That's the reproduction factor. The original reproduction factor of the SARS-CoV-2, back in the beginning of 2020, was somewhere between 2 and 3. And I've seen, low as 1.9, high as 3.1, so call it 2, 2.5. In contrast, Delta has got an R0 of somewhere between 6 and 8. That puts it on the scale of chickenpox. We're talking about really infectious. In fact, to find something that's more infectious, you got to go to measles, which has an R0 of between 11 and 13. Measles is one of the most infective agents we know, period. Most rapidly spread viruses that's been described to date, pretty much. All right. The other variants like Lambda and Mu are important. They have interesting mutations. Some are also more transmissible. Some cause more severe disease. But the fact is, Delta is so much dominated North America as far as the prevalence goes that while epidemiologists and infectious disease specialists keep their eyes on these other variants, they're not playing much of a role in disease as it occurs in the United States. All right. I know some people have been asking, well, what are the symptoms like? Well, interestingly, the symptoms haven't changed all that much. What I've got here is a little table that shows symptoms of patients over 18 versus symptoms of patients who are 5 to 17 years. And you can see they track reasonably well. There are some differences. First thing, everyone seems to experience a loss of taste, which is very much keen to the loss of smell. So that difference between the two is really nothing. Then that's followed by fever, but then it starts to diverge. In older patients, you see a persistent cough. What's interesting is you don't see that in younger patients. They might have chest tightness, maybe a sense of shortness of breath, but they don't have a persistent cough. Both groups exhibit chills. Both groups exhibit a loss of appetite. And then in the younger ones, you see muscle aches, which you also see in the older population. But the other thing that you see in the older population that you don't see in the younger patients is fatigue. So the persistent cough and fatigue in older patients are what distinguishes the disease between that which you see in the 5 to 17 or 18-year-old group. All right. Vaccinations, we know what vaccinations are. And I put this up again because now we're faced with, most of us, as Andy said, we've got pretty good vaccination rates. They're not what they should be, obviously, but I just pulled some stuff off of Johns Hopkins. In the United States, if you take all people, including those that are not eligible for the disease, 68% of the population has received one dose, 59% is fully immunized. If you look at the 12 and up range, those numbers go to 79 and 69 respectively. 18 or older, it's 81 with at least one dose and 70 with fully vaccinated. And if you go to older folk like myself, 65 and up, you got 99% have had at least one disease and 86% are fully immunized. So it could be worse. It's not great, as we'll talk about later. It could be worse, but a lot of people talk about herd immunity. And the problem is with these variants, that's becoming an increasingly elusive character. At some point, there's going to be enough vaccination and or enough experience with infection that an equilibrium will be reached. Not too dissimilar to flu. I don't think you're going to see the antigen shifts like you see in flu every year, but there's going to be some sort of equilibrium. What I'm meaning is that we are unlikely, at least the foreseeable future, to achieve true herd immunity. I don't see it. Where the bug would just disappear. For example, like has happened with polio, or like has happened with, it was measles until recently, what's happened with smallpox. In these cases, you got vaccination, like in the case of polio, was so effective that the disease burned itself out. There's really you can't find it in North America anymore. I think it's unlikely that we're going to get to that stage, at least in the foreseeable future, with the COVID strains. All right, so if we look at the variants, how well does the current vaccines work against the variants? Right, and this is one of my first big take-home lessons that is often misrepresented in the popular media. The goal of the current vaccines is not to stop the spread of SARS-CoV-2, certainly not directly. They weren't designed for that. Rather, they were designed to prevent severe disease, hospitalization, and death. And people get this wrong all the time. We were very fortunate in both the polio vaccine and the smallpox vaccine and the measles, mumps, and rubella vaccines, where they pretty much largely are quite effective. But in comparison, the influenza vaccine, if you got a good year, you might hit 50% efficacy. You might. Generally, it hovers around the high 30s and the low 40s. But the real take-home lesson is that the vaccines are extremely efficient. at preventing severe disease, hospitalization, and death. All right. When we look at the current vaccines against the original strain for which they were developed, you see this high efficacy rate, 95% for Moderna and Pfizer, 70s for Janssen and Janssen, 80s for AstraZeneca, 90s for the new Novavax. However, the data out of Israel, now Israel's an interesting case. You've got a small country, as countries goes, fairly homogeneous in its population. Everybody gets vaccinated. And what they saw was that there seemed to be a decrease in response, looking at antibodies. So the antibody response seemed to decrease over time, in particular when you look at antibodies that can recognize the variants. You have to be a little careful about just looking at antibodies. People look at antibodies because they're easy to measure. But there's a whole other side of the immune response, which are the T cells and the cell-mediated immunity, which is very long-lived. If you look at that, you'll find in many of these cases that the duration of your protection is longer. It just takes a while for these cells to become activated upon re-exposure to the virus, and then your antibodies come back up again, as anyone who has a booster can tell. However, Israel is not the United States, and only the Pfizer vaccine was used. What's happened in the United States? All right. Well, first of all, I should talk a little bit about the boosters before I get there. What the boosters are, and really at the moment, there's only two that are really available. Pfizer's has been approved. Moderna's is pending approval. We don't know much yet about the J&J, but we do know people that receive the J&J vaccine need a booster. But essentially, these shots are essentially the same vaccine as the original vaccine. So these are not tailored to the new variants. They're essentially recapitulation of what we've already seen. In the case of Moderna, they realized that they could reduce the dosage and still get a very good immune response. Pfizer is essentially repeating the same dose that they used originally. One of the questions one has to answer is, well, why get a booster? Well, if you look at the Israeli data, you would say, well, antibodies decrease over time. We should get a booster, but antibodies decrease over time always. If there's no virus around to stimulate the immune system, vaccinated or naturally immune, the antibody levels will go down. They come back up again upon re-exposure. All right, so, but what Israel did see in their experience was that giving the boosters did cause a boost in overall immunity to the virus in the state of Israel. The booster got up to protection from infection and hospitalization about 91%. 4 months later, it's still 77%. After the boosters, it all raised back up to about 90-some%. The boosters are now available. They're widely available. And then finally, which I'll talk about a little later, there's the concept of mix and matching. That is, there's nothing written in stone that says, if you got the Pfizer, you must get a second Pfizer. And in the case of people who receive the J&J, it is probably a better idea to get a booster of something like the Moderna or the Pfizer to get a much more heightened immune response. All right. But getting back to this declining efficacy of the viruses, in studies in US and China, looking at effectiveness. Now, we're not looking at antibodies anymore. We're looking for two things. We're looking at the ability of the original vaccination to prevent subsequent infection, which we call breakthrough. And we're looking at the ability to keep us out of the hospital. And looking at those Canadian and US studies, we see that the protection against infection has fallen for both the Pfizer and the Moderna viruses. It's true. You look at the breakthrough infections, people who are fully immunized, you see how many of those get infected. And what you see is that it's coming down from that original 90-some% down to 70s. However, what is very heartening is that the efficacy of the vaccines against severe disease, hospitalization, and death have held steady. even after several months, Pfizer and the Moderna virus vaccines remain highly effective at preventing hospitalization. But we got to keep in mind that with the decline in vaccine effectiveness for infection, we're still liable to see cases increase. Hence, the booster may not be such a bad idea. And as I said before, CDC has a success that if you got the single dose of J&J, you really do want to see about getting boosted with one of the RNA viruses. All right, if we look at this graphically, this is what we're talking about. All right, so in this plot, we've got 3 studies. We have a US Pfizer study, and then we've got a Canadian Pfizer study and a Canadian Moderna study. What we're looking at is here's efficacy against prevention of infection. And these are weeks, 10, 20, 30, since the second dose. And what you can see is, yeah, this shows why you have to wait after your second dose of your initial immunization, because it takes a while for the thing to get up to speed, and there it gets up to 90. Then here's what changes. In the case of Pfizer in the United States, the protection against infection did drop to around 60, low 60s. Interestingly, though, if you look at the same vaccine in Canada, which is a smaller population and more homogeneous, you don't see nearly as much of a drop. Still stays above 75%. Moderna's retains theirs a little even better in Canada. That's infection, protection against infection. However, when we look against protection against hospitalization, we clearly see that both vaccines retain their ability to prevent hospitalization quite effectively. Even weeks, we're out now 25 weeks in the case of the Canadian Pfizer study, 25 weeks after that second initial dose, they're still extremely effective at preventing people from having to go to the hospital, preventing severe disease. All right, looking at the boosters, who's eligible? All right, this is a big deal right now. The current guidelines coming out of the CDC are that people 65 years of older or younger people under 65, but who have significant medical conditions or have a frontline job status, they're first responders, they're health providers, healthcare providers, anyone on the front lines, these are eligible for the boosters. And in particular, for those over 65, getting the booster is very important. It provides that extra, as Rosenberg of the New York Department of Health says, gives you that extra protection, because in the older patients, the consequence of the disease are so significant. All right. The question is, what about everybody else? And what I'm seeing lately is state by state, states are opening up booster availability to basically everybody below 65. All right. When can I get a booster? Well, under normal circumstances, 6 months after you get either the second Pfizer or the Moderna shot. But in the case of patients with compromised immune system, I'm going to talk about that in the next slide, you need to get it much sooner, 4 weeks after the shot. Why? Because their immune system's compromised. They don't respond to the vaccine as well. And in the case of people with organ transplants, not at all, it seems, in many cases. All right, so the CDC recommends for those with severe immunocompromise 4 weeks after the second shot with an RNA vaccine. CDC is now recommending that people with the J&J should get a booster 2 months after their initial J&J shot. All right. Seniors, as I said, most likely to be affected by the waning vaccine immunity. And this is a twofold. One, their immune system isn't as good. And two, seniors in the United States were generally the first to get the shots. So it's been farther along. And as a result, the immunity has decreased with time. So that's why it's so important for seniors to get it. 71% of people over 65, it's about 36 million, completed their initial vaccination period greater than 6 months ago. And so far, only 31% of that population over 65 has gotten the booster. All right. This graphic basically shows that time from initial immunization times 0 here. These are the different age groups over 75, 65 to 74, 50 to 64, 40 to 49, 25 to 39, et cetera. And it shows that the over 75 got them first. Ooh, what did I just do? And then followed by the 65 to 74 group, 50 to 64. So you're farther out in time. So this is an added incentive for older patients to get the booster now and kick that immune system back up again. All right, who are the high-risk individuals? Well, as I said before, those at most risk are cancer patients or transplant patients, people who are on immunosuppressive drugs. In the case of cancer, it's the actual drugs that are used to treat the cancer. In the case of transplant patients, obviously, it's the drugs that shut down the immune system so they can tolerate the graft, that the graft will survive. In addition, people with chronic kidney disease, COPD, Down syndrome, heart conditions, obesity and severe obesity, sickle cell anemia, severe smokers, and type 2 diabetes, like yours truly, have some degree of immunocompromise and really should be candidates for the booster. All right, mix and match. Mix and match was originally poo-pooed by just about all the experts until people started actually looking at it and said, well, might be something here. And the something here is that no vaccine is identical. You know, each vaccine is each type of vaccine is different than someone else's vaccine, and your body benefits from that difference. Mix and match. And what a study out of NIH did, pretty big study, almost 460 patients, showed no increase in adverse events if you mixed and match, which is just like the flu. Every year, we don't remember what flu shot we got the prior year. We often mix and match with flu shots. And in some cases, we can do a whole lot better. As I said, the J&J primary shot, just about anything else you get after that's going to help you. As a matter of fact, if you look at people who got the Moderna following the J&J, they got the greatest increase in immune response, followed by Pfizer, and then followed by a second J&J response. So in the case of someone who got the J&J response, it's highly advisable to go out and seek that Moderna shot. And if you can't find a Moderna, the Pfizer. And both the FDA and the CDC have now pretty much said, yeah, this is a good idea. Nothing wrong with it. All right. Here's another big point, though, I really want to make. While the boosters are important, getting the shot into unvaccinated people is so much more important to really slowing or stopping the spread of COVID-2. I cannot emphasize this more. The most recent data out of Johns Hopkins says there's about 60 million eligible people in the United States that are unvaccinated. And it's been said by more than a number of people, including Tony Fauci and guys like William Shatner, that right now we're looking at a pandemic in the United States of the unvaccinated. Mike Osterholm said that this condition is akin to a tremendous amount of tinder for the corona wildfire to burn. He's absolutely right. When you have large pockets of vaccine hesitant people, vaccine denying people, just unvaccinated people, the virus can get in there, especially something like Delta, whip through it. And what happens is the more viral spread, the more chances we're going to see new variants emerge. The virus doesn't replicate, it can't mutate, we can't see new variants. All right. And as Andy said in the beginning this evening, he said, after falling for 7 weeks, unfortunately, the infection rates are starting to are increasing again here in the Northeast, in the North Midwest, and in the Northwest. They're going up. It's not so simple. There are pockets now that were ravaged during the summer, like the South, are having remarkably low infection rates right now. I wouldn't hold my breath on this. Is there going to be a fourth wave this winter? I hope not. We need to protect against that, but it's a possibility. So that brings us then to the next issue. What about vaccines in kids? All right. The Pfizer vaccine has now been received emergency use authorization in May, and is now has EAU for patients 12 to 17 years. Moderna is finishing up their study. If they don't have it right now, I just looking around this morning, I couldn't see if a final word came in. They're going to get it very soon. J&J started their 17 to the 12 to 17-year-old study in April. I don't know what the results are of that yet. In addition, we've heard talk in the last couple of weeks about a vaccine for very young children, 5 to 11 years. And data has been presented to the FDA from Pfizer on this. As a matter of fact, there has been pilot vaccination programs in both New York and New Jersey schools. They were quickly wiped out. The demand far exceeded the supply. We don't have formal approval yet, but it's going to be imminent. Israel did give approval for the Pfizer vaccine in the 5 to 11-year-old group. And so far, there have been really no reports. I'm going to talk about that in a minute. Reports of any blip in the adverse event profile in these very young patients. I'll just state personally, I think if I have a 6-year-old grandchild, she's already got her first dose. She can't wait to get back to school. She will get her second. And then several weeks after that, she will go back to school. That's my own personal opinion on it. All right. This all raises the question, as I posed, do these vaccines really block transmission? Still a big question. The results are starting to come in, but it's still not certain that it's going to truly block transmission going down the road, in particular in the light of Delta. And for that reason, I don't think we can really let up on masking and social distancing. Fact is, the country is, for the most part, open right now. But if I'm going into a crowded environment where I don't know all the people there, like let's say Food Town or ShopRite, I still put on my mask. As I mentioned before, the recent data from Israel suggests that the boosters do decrease transmission rates. But as I said, the viruses were designed to keep you out of the hospital and keep you from severe sickness and death. They weren't really designed to block infection. However, if we get enough people vaccinated, we're going to really slow the transmission down, and an equilibrium will indeed be reached. All right, what about side effects? Well, there's been talk recently, of course, about the cardiovascular side effects, and they're still rare. A new side effect, though, was described with the RNA viruses, described in France. And it's basically a sudden shoulder pain followed by paralysis. Not well understood. They've seen it in some flu vaccines as well. But I got to tell you, it's really rare. In the United States, there have been 6 cases reported in patients 19 to 69, up to 50 days following the Pfizer vaccination. 3 patients were after the first dose, and 3 patients were after the second dose. And that's in the tens of millions of patients who have received the Pfizer vaccine. In the case of Moderna, there's only 2 cases been seen. There have been 8 in England that received the AstraZeneca that they've seen these. I don't know what to make of this. It's an interesting blip. It's a severe side effect. They seem to go away, but it's exceedingly rare. But it is out there. You will hear about it. All right. If we look at the more general safety profile in the 12 to 17-year-old, we have more data. In the 12 to 17-year-old population, about 6 million doses have been given out as of the end of August. Of that 6 million doses, there have been 591 potential adverse events, of which 35%, so 35% of 591, a couple hundred, were considered serious. These include reactogenicity. I love that thing. It means I got a reaction to the vaccine. General malaise, I don't feel good. Post-vaccine discomfort, followed by, and these are more important, myocarditis and pericarditis. Myocarditis, inflammation of the heart tissue itself, pericarditis, inflammation of the sac surrounding the tissue surrounding the heart. But as you can see, these are 16% and 7.2% of the 35% of the 591 out of 6 million. So we're talking about extremely low incidence. Of the 33 patients with myocarditis, 40% developed it after the first dose, 54 after the second dose. Of the 15 with pericarditis, 53 after the first dose, 40 after the second. There's also been 3 cases in this population of multi-system inflammatory disease, which is a very serious disease. You get this large, it's like a cytokine storm, and it infects multi-organs within the patient. But so far in the 12 to 17-year-old, we've seen 3 cases out of 6 million. So again, it's a very rare side effect, very rare. All right. Now that's about all I was going to say about vaccines. It's also important to note that there are now finally emerging therapies that are not vaccine and can be used and administered to people shortly after they develop symptoms. Many of us, I'm sure, know about the monoclonal antibodies. They are by Eli Lilly and the Regeneron Corporation. President Trump got the Regeneron cocktail of monoclonal antibodies, and I, for one, think that he may have a lot to owe the Regeneron Corporation for that. I think it really got him out of the woods. He later would get vaccinated, but these things are not, they're expensive. They're about over $2,000 a treatment. It's an hour-and-a-half infusion, IV infusion. It must be done in the clinic. So this is not a simple thing that you can just bop down in the CVS and buy. On the other hand, first Merck, and then most recently in the last week, Pfizer, have introduced drugs, true dyed-in-the-wool pharmaceutics, that do have significant effectiveness against the COVID infection. Let's talk about the Merck. That came out first. It's been approved formally in the UK. It's got a preliminary approval by the FDA. It's inexpensive. It's about $700 per patient. You take a few pills. There's 4 capsules twice a day for 5 days. All right. However, it was shown that this Merck drug, Molnupiravir, could reduce hospitalizations and death by about 50% in unvaccinated adults that are symptomatic. Paxlovid, which is Pfizer, I don't remember if you might remember that Pfizer came out about 20 years ago with another antiviral called Tamiflu. It's on the same class. It's a protease inhibitor. It's important in that what it does, the drug basically inhibits an infected cell from assembling the virus. It stops the viral infection cold. And in a study involving over 1,200 patients, it showed an 89% reduction in hospitalization and death when given within 3 days after the start of symptoms. And this is very significant. The ethics committee stopped the trial early. I don't know if you've run into this before, but when you see that, that's of note, because that means that the treatment arm was doing so much better than the control arm that the ethics committee said it's unethical to keep giving people placebo. The benefit is so slam dunk clear. And they did. They stopped the Pfizer study a little over 1,200 patients into it because the efficacy of Paxlovid was so remarkable. It's geared to older patients, basically over 60, and high-risk patients like the Merck. It's got a few less pills, about 30 pills given over 5 days. But these, and they give it with another drug called ritonavir, which has long been used in the HIV community. What ritonavir does in this case is it's not an antiviral in itself, but what it does is it prolongs the drug, the Paxlovid, within the patient. It prolongs the half-life of the patient and increases its efficacy. This is remarkable. This is something that we really need to keep an eye on because while it may not be a get out of jail free card, if you've got a high risk patient, even if they've been vaccinated and they develop symptoms, this does look like it has the ability to stop the virus pretty effectively. All right. How are we doing, Andy? Oh, I've already used up a lot of time, haven't I? I got to wrap this up. All right. I'm going to talk a little bit about long COVID. This, I got to tell you, is a disturbing field. It's vexing. There's so much we don't know about it, but there's a massive need. These people are real. This disease does appear, and it's difficult to treat. Essentially, we know that the virus has like several mechanisms of action by which it can make you sick. Clearly, it can infect susceptible cells. For example, in the epithelium of our lungs, our sinuses. It can infect those cells and kill them. It's a regular virus, lytic infection. It can also cause an acute inflammatory process, as we see in SARS pneumonia. And that inflammation can kill you. As a matter of fact, in the 1918 and 1919 influenza pandemic, that was the major mode of action of killing young people. Their immune response was so overwhelming and their inflammatory response, their acute respiratory distress, so rapid that it was killing people within hours. They had healthy immune systems. Virus killed them because of that healthy immune system. A third way, which is common to almost all respiratory infections, is a secondary bacterial infection. You get the virus, you get a respiratory infection, it decreases your immune system, it compromises the lung integrity, and you get a secondary bacterial infection, and that's what gets you. That's common. However, there is a fourth mode, which we know SARS-CoV can work, and that's to cause chronic inflammation. Seems to do this through microclot formation. The target of the SARS spike protein is a receptor on almost all cells called ACE2. ACE2 is very much involved in blood pressure regulation.
I found this on the web.
Oops. Sorry about that. And what happens is the virus attaches to this ACE receptor on non-susceptible cells, and it doesn't infect them and then lyse them. But what does happen is, as a result of that binding, the immune system sees it and a microclot forms at the site. And it's that microclot that basically triggers a much longer standing chronic inflammatory response. And this can happen in the gut. It can happen in the heart. It can happen in the liver. It can happen in the brain. It can happen in other tissues like the skin. And so what is believed is that this non-lytic viral binding to ACE receptors in a variety of tissues, in some, clearly not all patients, but some patients, can lead to a chronic inflammatory response that's essentially initially local, and that can give rise to many of the symptoms that are now associated with long COVID. As a result, much drug development nowadays is focused on what we call bifunctional compounds. Compounds like that nice Pfizer drug that actually shut down viral replication. But then the other functional is to selectively inhibit the inflammatory response. Now, you can't just willy-nilly do that. For example, there is a drug that's used for very severe patients called tamoxifen. Tamoxifen is a very potent corticosteroid, but you have to be very select about when you give it. They generally give it to patients who are either just getting on a ventilator or they want to get them off, but they've already got a severe infection. Their immune system is already well compromised. They need to stop the inflammation to save the patient. All right, so you go in there with a strong corticosteroid. If you were to do it earlier, you run the risk of shutting down the immune system completely, and then the patient rapidly succumbs to the virus itself. So that's like the wrecking ball. You use that when you have no other choice, and you've got to stop the inflammation to save the patient. But it's not good if you're just symptomatic or you're moderately symptomatic. So new drug development is focusing now on the development of more selective anti-inflammatory treatments that can shut down this chronic local inflammation and stop things like the long-term GI symptoms, like chronic diarrhea that just doesn't go away, or brain fog, or myocarditis or pericarditis. But it's a vexing issue, and there's a lot of research being done on it, and there's a tremendous development to come out with new combined modality therapies that do address this. All right, so that's at the moment all I'm going to say about long-term COVID. I know one person asked, if I was asymptomatic, or all by extension mildly symptomatic, is it possible that I could come up with long COVID 6 months later? Unfortunately, the answer is yes, it is. It's not terribly common, but it does happen indeed. All right, the last thing I want to do is talk a little bit about misconceptions, and I'll end it on here. These are some recent ones I pulled off, and yeah, I've got a little tongue in cheek on it, but the fact that these are out there and that people believe them is a very serious issue. One I heard recently is that Colorado's governor issued an executive order that allowed hospitals, when approaching or achieving complete capacity, like let's say they have no ICU beds, they can redirect patients to other institutions that have more capacity. The social media networks basically took this and said that the governor said that hospitals can refuse to treat patients who are unvaccinated. The governor's order says nothing about vaccination status, but that's how it's been interpreted. Another one that was good is Albert Bourla, who is the CEO of Pfizer Pharmaceuticals, has been arrested and charged by the FBI with fraud for his role in deceiving customers on the effectiveness of COVID-19 vaccine. This came from a Canadian website, quoting an unnamed FBI agent. Well, as I showed you, stuff's happened. The bug that we had in the beginning of 2020 isn't the bug we got now, but it's still awful effective against keeping you out of the hospital and keeping you from dying. I don't think the FBI has arrested the CEO of Pfizer for misleading the public. Another one's a video clip that shows Vice President Harris saying virtually everyone who has been in the hospital sick with COVID right now is vaccinated. Actually, she said the exact opposite, and they doctored the video. Another good one I heard, and we have to be serious about this because it's such a horrible event, but a QAnon source basically had a false claim suggesting that the vaccines were responsible for the fatal crowd surge at the Travis Shoot concert in Houston, and implied that the magnetic frequencies in the music compelled concert goers to stampede toward the stage due to the graphene oxide in their systems from the vaccine. A couple problems. One, none of the vaccines use graphene oxide. Two, graphene oxide is not magnetic, nor does it even conduct electricity. Oh well, try again. Then you may have heard that there's a report going around that 3 giraffes at the Dallas Zoo died from the COVID infection. They hadn't been vaccinated at all. And then finally, one that I put in there. One of the problems that we are seeing with vaccine hesitancy or even vaccine denial is this concept of invincibility. People think they're healthy and they can't get the disease. And time again, studies are done, questionnaires are given. People go out and ask, why don't you really want the disease? And says, I'm healthy, I'm not going to get it. And this is turning out to be a huge, huge barrier to effective vaccine, to cut into that 60 million eligible people who are still unvaccinated in the United States. And with that, I thank you for your time. Now I have to unshare this, right, Irene?
Yes, please, you may. And thank you, sir. Thank you very much. We do have some questions that I'll read from the chat. If you do have any questions, please put it in the chat. David Wilson, I know you started off here. Did Dr. Sean answer your questions during the course of his presentation?
Let me unmute myself.
Hey,
Okay,
You're on there. You're on there, Mr. Wilson. We can hear you.
I think the answer is Moderna booster is my wife's choice. Does she have to wait for that?
How long ago did she have her last vaccination?
6 months, greater than 6 months.
Oh, she should take it tomorrow.
All right, so any version?
There's only one of the Moderna, and if she can't get the Moderna, I would recommend Pfizer, even if that's what she got the first time. But if she can get the Moderna, if she's 6 months out of her last dose, she absolutely should get it.
All right. Also, 5 to 11-year-olds had Pfizer shots a week or so ago. How long should they wait before they get?
They need to wait. If it's Pfizer, they need to wait another 2 weeks. 4 weeks general is the time between shots with the Pfizer.
2 to 4 weeks. Okay, and for my granddaughter,
Mm-hmm.
is less than 5 years old, 2 daughters. Both have got a shot earlier.
They're less than 5 years old and they got a shot.
They do have a shot.
Hmm.
It's not clear which one they have, whether it's a fake one or a real one.
We don't have data. There's not a lot of data on vaccine administration to children under 5. So, Mr. Wilson, I don't know how to counsel you on that. I would strongly suggest you talk to their pediatrician.
All right,
Talk to several pediatricians and see what they think.
I'll do that.
Yeah.
That's it.
Okay. Thank you, sir. Okay, Darlene, you had a question. An unvaccinated friend asked me why an unvaccinated person is required to get COVID testing for work, but vaccinated people don't. They question this since both vaccinated and unvaccinated can spread COVID.
She's got
Is
a
there-
point. Oh, I'll say she's got a point. Unvaccinated, it's obvious. You know, they could be sitting there with an occult infection, you wouldn't know it. But as we do know, especially with that incident in Provincetown, Cape Cod, vaccinated people can be, at least for on the order of about 5 to 10 days, remarkable spreaders of the virus. What seems to happen is the virus, one of the things that people don't understand, viral infection is not an all or none thing. It's dose dependent. And you can be extremely well vaccinated, and if you get a huge dose, you're going to get the disease. Hopefully you get a mild disease. My brother had 2 Pfizers and a booster, and he went to teach school in the Racine, Wisconsin district, and there were 2 kids that had real whopping infections, and darn if he didn't break through. So if you get hit with a strong dose, what happens is the virus can hang out in your sinuses. And it can be happy there because the vaccine that we're using is a systemic vaccine. It's not a nasal vaccine. So that means we're immunizing the whole body, and it's going to take a while for the body to respond to such a local infection. What it means is that those people don't necessarily get sick. In the Provincetown, there was like 100 people involved, and a couple of them got moderate disease. No one ended up in the hospital, but they were wonderful spreaders. Why? Because the virus was hanging out in their sinuses, their nasal cavities, and they were thus spreading it, even though the rest of their body was well protected by the vaccines. So the lady makes a good point. Why don't they require vaccination? I can tell you, I traveled to Brazil earlier this year, and I was fully vaccinated. They still required us to they wouldn't even accept the rapid antigen. We had to get the PCR for the same reason. So I think there's a good case. If it's a sensitive job, even though they're vaccinated and they have documentation, you still may want to insist on a rapid test.
Okay, thank you. Darlene has a follow-up question. Is there a difference between cell memory, herd immunity for unvaxxed people who get COVID versus vaxxed people?
It's not as good. Yes, there is a difference, and it's not as good. They do have cellular immunity, but they have been looking at this. They've looked first at antibodies, and they started to look at cell memory, cell-mediated memory. And what they were able to show was that on that scale, people who had a mild to moderate or worse disease had X as their recall antigen status, their ability to respond to the antibody again. The next highest group were those that got fully Pfizered or fully Moderna, but the best protected were those that had an initial infection and then received one dose of either the Pfizer or the Moderna. They had the most significant protection by both humoral immunity and cell-mediated immunity as determined by the reactivity of their memory cells. Does that answer the question? In other words, just having the natural immunity doesn't necessarily, natural exposure by infection doesn't necessarily buy you the best immunity.
And hi, this is Darlene, and that was the question, the difference between the cell memory. I'm vaccinated and my friend is not vaccinated, and they're saying, well, I've had COVID, so I have the antigen, I have the antibodies in my system, which is a natural, versus getting this stuff injected in me, basically.
But if my presentation today said anything, the viruses have changed. And the infection she may have had might have been the original COVID, and that's not Delta. So the idea is that you can only respond to what you've been exposed to. And in the case of these modern RNA vaccines, as well as the J&J, the AstraZeneca, and the Novavax, is they try and load them up with different antigens to address this issue so that they're multivalent. They are seeing not only different strains, but they're seeing a number of different aspects of the virus. And that generally gives you better immunity than just getting the infection itself, which is going to be a snapshot at the time you got infected.
Great, thank you very much. I'll just, if you don't mind, Andy, just real quick, I had thrown another thing when you were talking about misconceptions. This same person says, and others like them, that COVID's not going to go away as long as Big Pharma is making millions and millions and millions of dollars off of all of this. This is all due to the pharmaceutical companies getting rich, and it's not going away. So.
I don't appreciate the cause and effect. I understand the sentiment, but I don't see the cause and effect there. How are the pharmaceutical companies promulgating the virus?
They're not promulgating the virus. They're promulgating the forcing of people to get vaccinated and breaking.
And that extends the viral infection.
So they're just feeling their civil rights are being taken away because Big Pharma is making millions of dollars and the government's being led by Big Pharma.
Please don't think I'm facetious, but that's a little like the old thing, do you walk to school or carry your lunch? It's a non sequitur. Those two don't mix. Yes, the pharmaceutical companies are making money off of it, but they are not. I have yet to see anyone give me an example where any of the pharmaceutical companies are doing is extending the pandemic. You're extending the pandemic by requiring people to get vaccinated. That's a breathtaking example of non-logic.
Okay.
Agree
Thank
with you. There's just no convincing.
Oh, of course not. Logic doesn't work. That's right from the Bible. It's right from the Bible. Logic won't work, of course.
Okay, Gail has a question. Do you see a need, Sean, for senior population to require a booster every 6 months?
That's a good question. I wish I could answer that question. I don't know. We have to see how this goes out. What happens with a booster? All right, you've already got the memory cells circulating in your body. You've had 2 shots of the original shot. So you've got memory T cells and memory B cells. They mostly hang out in the bone marrow. They may hang out in the spleen or the lymph nodes. And But as I said, the antibody levels rise and they fall. You don't want antibodies there all the time. People forget that. That's not a good thing. It's like always having your AK-47 on the table, locked, loaded, and with a big clip in it. That's not a good idea. So the question is, 6 months from now, in order to maintain good protection, are we going to need to do this again? And there's not enough known about this virus to answer that question. It's a great question, and I wish I had a good answer.
Okay, Jessica has a question, two questions. If a child gets COVID, even after being vaccinated, should we be concerned about long-term effects COVID?
Great question. It's something people are looking at, and we don't have enough experience yet to say. COVID in kids is different than COVID in adults. I think I showed some data to that shows some of the differences. And this is a real good question. A careful answer would be in some small percentage of the population, the answer is probably yes. What is that fraction? I don't know. But from what we've seen so far about this virus, it's sneaky. And as I said, this bit with microclot formation is disturbing. One of the things to note, though, about the kids is they do have a much more robust immune system than we do. And so that if they are properly immunized, they stomp on the infection. If that wasn't the case, the polio vaccine wouldn't work. The MMR and the TPAP wouldn't work nearly as effective as they do. They work well because young people have a very robust immune system that when exposed to the antigens in these vaccines, you get a very whopping, long-lasting response. And we're confident that for the most part, we're going to see that in the kids. That doesn't mean you can have the occasional child that does show long-term sequelae. But what the nature of that is and what the frequency is, I couldn't possibly answer it right now.
Okay, similar, following up regarding children, Jessica says, I have a 6-year-old and a 2-year-old. Would you trust putting them into a classroom of mostly, if not all, unvaccinated children, even after they get vaccinated?
Were they 2 and 6, did you say?
Yes.
If the 6-year-old was fully vaccinated, I'd let him go, but I would really try and teach that kid to be careful as much as you can teach a 6-year-old. The 2, no. I would hold the 2 home until she's old enough or he's old enough to be vaccinated and protected.
Thank you. Okay, let me see. I think that's all the questions. Am I missing any questions?
No, thank you, Sean, for your time and your effort.
Well, thank you for your excellent questions, David. Much appreciated.
I'd say, Sean, thank you for your research, your expertise, your thoughtful consideration, especially to the very personal questions. We appreciate you speaking scientifically as well as in a plain spoken manner.
I hope it was clear. I should say where I'm speculating more. I try and indicate that, but you're right. But this is the real world, and sometimes you have to ask, all right, what would you do? Well, you got my answer.
All right, sir. Well, wish everybody a happy and healthy Thanksgiving.
Amen.
As we go into our holiday season. So thank you all for joining us. As Irene
Thank you for
posted,
having me.
there will be we will be posting the video. You see the link there if there's anything you want to miss or pass along to others. Thank you again. I wish you all a good night.
Bye-bye now.